Hi, this is your biology tutor Isolately. Our lesson for today is osmosis in potatoes, right? So, straight to the point, what is it that we want to achieve by the end of this lesson? We want to demonstrate the process of osmosis, all right? Osmosis with the O, okay? We want to demonstrate the process of osmosis. We want to show that water actually moves through our plant cells from a region of high water potential to a low water potential. Now, let me differentiate osmosis from diffusion. I noticed that in my previous lessons, I was somehow interchanging the words. So, osmosis, this is when you are mostly dealing with water, right? So, osmosis, it's the movement of water molecules, right? Underlining the word water molecules. So, this is water that is moving from a region of high water potential to a region of low water potential through a partially permeable membrane. Whereas diffusion is the movement of molecules such as gas molecules, right? Now these molecules, they are moving from a region of high concentration to a region of lower concentration down a concentration gradient. All right, so that is the difference. I'll actually make a presentation on this as well. But anyways, proceeding with our lesson for today. um Going to our experiment, we need a potato, or rather we need potatoes, we need a containers, and then we need a pin, we need pure water, or rather tap water, and also we need a solution. We already defined what a solution is in the previous video, which you'll find in the description box below, all right? And then when we have all these, we are good to go with our experiment. Now, what should we do to our potato? Our potato, we need a knife, and then from there we need to cut out, we need to make a hole as it's shown in the pictures in our presentation, right? So we need to make a hole, and then after we are done making that hole, we add in water. It's either we are adding water in our potato, or rather we are adding a solution, and then this potato has to be in a container, right? And then the function of this pin is we want to put the pin where our water level is ending. All right, so the function of the pin is we want to put the pin in our hole to show the starting level of water, or rather the ending point of our water. We want to see, will the water actually increase in our potato, or rather will it decrease? Okay, so you understand this when we move on to the next slide, which is next. All right, so what we have here, we have potato. This is our beaker number 1, and then this is our beaker number 2, or rather B. Now in beaker A, beaker A contains our first potato, which is potato A. Now what is it that we are finding in our beaker? In our beaker we have pure water, or rather tap water, all right? And then from there we have our potato, and then from our potato, inside our potato, in the hole that we created, we are adding salt solution. If you are dealing with salt solution, that is fine. You can also, instead of salt solution, you can actually get a sugar solution. All right, so that is what you are getting in potato number 1, and then you are putting a pin to show that this is where your salt solution is ending. And then moving on to our beaker number 2, where we are finding our potato B, we have in our beaker, instead of having water, we have salt solution. Whereas in the potato itself, we have pure water. All right. So the difference between these two beakers is that in potato number A, in potato A, not potato number A, goodness. Okay, so in beaker A, you actually have pure water, or rather tap water, whereas in beaker B, you have salt solution. Whereas moving on to our potatoes, in potato A, or rather beaker A, you have your salt solution. whereas in potato B, you actually have your water, or rather your tap water. And then in both our potatoes, we have a pin to show where our water, if it's water that we are dealing with, or rather our solution, where does it end? All right, so let's move on to our results. We want to see what will happen. Remember, we are talking about osmosis, right? So osmosis, or rather water, will actually let's start off with potato A before we go to our result. Let me just explain what happens. So what will happen here is that because in your beaker you have pure water, or rather you have water, whereas in your potato you have salt solution. Because of osmosis, water will move from the beaker to the potato. Why is it so? It's so because your beaker has a high water potential. This is water. Whereas in the potato, we actually have a solution. And by the term of osmosis, osmosis is the movement of water molecules. So this is our water molecules. It's the movement of water molecules from a region of high concentration. Where is water in higher concentration in this experiment? It's actually in the beaker, to where it's in low concentration, which is in the potato, right? This is why your water is moving from the beaker to the potato. Now, because water will leave the beaker and go to and enter the potato through a partially permeable membrane, you will see that the water level in your potato, it will actually increase such that your pin or rather the water level will be above the pin. All right, whereas in potato B, because our water is within the potato, whereas in the beaker, that is where our salt solution is, water will actually move from the potato where it's in high concentration, and then it will go through the potato where it's in lower concentration to the beaker. All right, and then what is it that you'll notice? You will notice that your water level here will actually decrease, whereas your water level in potato A will actually increase. All right, so your salt solution in your potato, in potato A, will be more diluted, whereas your beaker will have less water. Why is it having less water? It's having less water because water moved from where it's in high concentration to the potato, and then because it moved to the potato, our water level in the potato increased. Now, because it increased, that means that our salt solution will be more diluted. It won't be concentrated as it was initially. All right, and then moving on to potato B, because water is moving from the potato and then this water is coming to the beaker, you find that your solution, which is in the beaker, will be now more diluted, and then your water level in the potato will actually decrease. All right, so let's quickly go through the results now. This is the experiment. What do we need? We need to cut our potato, or rather we need to make a hole in our potato, as we saw in the previous slides. And then from there we need to put a pin. Why do we have to put a pin? We want to show the starting level of our water, right? Just as indicated, we want to show that, okay, this is where our water level is when we are starting with our practical. And then after we are done with our practical, we go back and see whether did the water level go above the pin, or rather did the water level decrease. That is the function of your pin. And then from there you are either adding water or rather a solution. So you are adding water to your potato, and then you add water to your beaker. So you are adding water to your potato, and then you add a solution to your beaker, whereas in the second case you are adding water solution to your potato, whereas you add pure water to your beaker. And then from there you allow the experiment to stand for at least 2 hours, which is minimum. It can be over, or rather we can go above 2 hours. All right, and then we are also told that we should always remember to cover the beaker to prevent the process of evaporation. We don't want our experiment to be affected by any other environmental condition. We want to be as accurate as possible. So because we want to be as accurate as possible, we want to cover our beakers so that evaporation will not affect our results. All right, we don't want water, or rather our solution, yes, we don't want water to leave either the potato or rather the beaker, and then yes, in short, we don't want our water to evaporate, because when that happens our results will be affected. We will not know whether it's osmosis which occurred or not. So this they are mostly likely to ask. So you should know, they can ask you why should you consider covering your beaker or rather the, whatever that you'll be given in your exam or in a test. So the answer to that is you want to prevent evaporation, okay? And then also you should know why do you need a pin. You are marking the starting level, okay? The starting level of your water. All right, so going to our results, in our results in potato A, let's quickly look at what is happening in potato A, and then we go back to our experiment. All right, in potato A, our pin, that is where our pin is, and then we can see that our water level actually increased. Initially, when we started with the experiment, our water level was by the pin, but after 2 hours of letting your experiment, or rather after 3 hours or whatever period you were asked to leave your experiment for, you find that in potato A, your water has actually increased, and then that is in the potato, whereas in the beaker your water will actually decrease. Now going back to the experiment, we want to see what is it that we have here. This is actually your salt solution, and then whereas in the beaker that is actually our pure water. All right, so as you can see, your water level was here, and then what is it that you have in your potato, or rather in the hole that you made? It's salt solution, whereas in the beaker it's water solution, and you can also see the level of your water in the beaker. Now what happened here? What happened is osmosis. So the process of osmosis took place here. How? This we already explained. We already said that water moved from the beaker into the potato through the cells of our potatoes, through a partially permeable membrane. So water moved from where it's in high concentration, which is in the beaker, and then it went through the cells of our potatoes, and then because of that our water level in the potato actually increased, making the solution to be more diluted. And how do we know that the water level increased? It's because of the pin. We know that initially the starting level was at the pin, and then after 2 hours now our salt solution is above the pin. All right, whereas in the beaker our water actually decreased. So this they can ask you, they'll ask you what is happening in the beaker, or rather what happened to your beaker after 2 hours, and what happened in the potato after 2 hours. So that is the explanation that you give. Your water level in the beaker decreased, whereas your water level in the potato increased. Now you need to reason why did it increase. It's because of osmosis. Moving on to potato B. Now in potato B, in our hole we had pure water, right? And then in our beaker, this is where we had salt solution. Now because by the term of osmosis, osmosis is the movement of water molecule from the region of high, where is it in high concentration? It's in the hole, right? So it's in the potato. So it's the movement of water molecule from where it's in high concentration to where it's in low concentration. So in the beaker we have less water as compared to the hole. What you have in the beaker is your salt solution, but rather not water. So this is why your water moved from the potato through the cells of your potato, which is our plant, and then to the beaker. Now what happened to the water in your potato? Your water level in the potato decreased. How do we know? It's because the water is now below the pin. So where your pin is, this was the initial level of your water. And then we can see that after 2 hours, your water level actually decreased to that. All right. Whereas in our beaker, our salt solution level actually increased. Now, this is what happens in terms of osmosis. All right. And then now let's interpret our results. What happened? We are told that in beaker A, the level of water in the container decreased, of which we saw. The level of water in the container actually decreased, whereas the level of our salt solution in the potato actually increased. And then because now the level of our salt solution increased, we know that our salt solution will be more diluted. Whereas in B, the level of water inside the potato actually decreased, and then because the level of water inside the potato decreased, the level of our salt solution in the beaker increased, making the salt solution which is in the beaker to be more diluted. Okay, now to interpret, those were our results. Now to interpret our results, we are told that salt water on the inside, it has a low water potential. Remember, we are referring, this should not even read as salt water. I'm sorry for that. This should be salt solution. So our salt solution has a less water potential as compared to what we have in the container. Now, because of that, water will actually move from the container where it's in high concentration into the potato where we have less water. And then because it's moving to the potato, you'll find that your solution in the potato will be more diluted. All right. And then whereas in B, our salt solution, which is the one now in the container, it has a low water potential. Sorry for that. It has a low water potential. All right. Now, because it has a low water potential, water will move from the potato into the beaker, and then because it's moving to the beaker, we are told that water moves from the potato to the container, or rather the beaker, by the process of osmosis. Now, because of that, we are told that the level of salt water, or rather the level of our salt solution, it will actually increase in the beaker. Now, because it increased in the beaker, what will happen to our salt solution? Our salt solution will be more diluted. All right, it's more like you are at home, you are making oros, you are making your concentrated oros. Now you are pouring a bit in a cup, and then after you want to add water, and then you notice that no, my oros, it's way too concentrated. What do? You take another cup and then you add water so that you can dilute it more. All right, so it's the same thing. Your salt solution, it will not be that salty anymore. Why will it not be that salty? It's because we have water. It's because we have water that is moving from here, and then it's coming into our salt solution. Because it's coming into our salt solution won't be that concentrated as before. All right, so this is why we are saying that it will be more diluted. Right, so that brings us to the end of our lesson for today. So please do not forget to subscribe. Also, if you are a subscriber to this channel, thank you so much. Keep on watching, and then if you know of anyone who is upgrading, or rather anyone who is in grade 12, please do talk to them, share the link. Let's do the thing. Let's get our straight A's and our B's. All right, you can also give your suggestions. If you feel like I'm too fast, you can always comment. You can always leave a comment, and then I'll try to slow down. If you feel like maybe there are things that I didn't explain properly, you can always as well comment. You know, it's a learning platform, so I'm learning from you, you learn from me. All right, so let's grow together. Let's do this. Thank you so much for your time once more. Again, we meet again. Salute.